Achievement actually is the main reason of happiness. So getting a message like this from patient with non-obstructive azoospermia, was last week in 21 September, make me really happy, and I am sure that all of you feeling the same thing when you success with some treatment that take a long time. So this is our main goal, to make people happy. And to get this achievement actually are the results of a lot of research, a lot of hard work. And the main goal of our research to find a solution that can help people. And I am sure when you visit a conference in different countries, you will see a lot of, you are witness of for a lot of development in the fertility field. All the new equipment, disposables, reagent, and all of that. So this field is keep actually develop, and we all the time find new things to increase the success chances in the fertility field. I'll give you an example. In the 70s, they developed the IVF method to help men with low sperm count, so they can have also babies. And of course, this method keep developed, and then 10 years later, we have the XC method that will help more men with infertility problems. In 1996, a new method called Prostate technique also was developed to help men with non-obstructive azoospermia with maturation arrest with round spermatid. Unfortunately, it was disappeared for 20 years because a lot of limitations. And in 2015, it came back to the research and to the clinical use. Still, there is a lot of challenges, a lot of limitations. And there is a nice article that published this year about this technique. If you're interested, you can take a look. What I want to say that we keep discovering new things to make and increase the success chances for couples. Azoospermia, also the same thing. Before 1990, there is no chances for men with non-obstructive azoospermia to have kids, babies, because there is no sperm. Then we discovered that the sperm actually inside the testis, we can extract it and use the IVF or the X method for fertilization. Again, after 10 years, new method was developed called the microtease, using a microscope looking for live tubes inside the testis to extract the sperm from there. So research actually is the heart of everything in the development and solutions. This is actually from 2015. Even it's old, you can see that the male factor responsible for 60 to 70% in fertility cases, and this is in the Middle East. And this percentage keep increasing every year. We don't really know why. There is a lot of reasons, maybe the lifestyle. There is a few published articles about that, but we still do not understand why the infertility in the male field keep increasing all the time. Because of this high percentage of male infertility, there is a high study subjects, around more than 100,000 published articles every year, and it's increasing. In azoospermia, there is more than 20,000 published articles around the world trying to figure out and to understand more about this process. Yes. So spermatogenesis is a highly researched subject, and the more we learn, the more we understand. And I can describe it like a puzzle. The more pieces we discover, the knowledge will be much better and more clear picture we can find. So let me take you a tour tour in our research that we have done for years. And let's start with the Spermatogonia stem cell. We developed a new method that called 3D culture method, and the main goal from this method actually to culture Spermatogonia stem cell in vitro. Spermatogonia stem cells are very difficult to culture. They need very special conditions, so you cannot just culture it in a flask. So we developed this method for two main goals. The first one is to develop a new method to understand the proliferation and the differentiation of the Spermatogonia stem cell. And the second goal was to have a solution for men with non-obstructive azoospermia, to take the stem cells out, do the culturing, get sperm in the dish, and then we can use it for eggs. We did actually, we took a Spermatogonia stem cell from 7-day mice. We got a single cell suspension, and we developed this method. It was very unique to our lab. It's 2 layers of soft agar. We have in the lower agar, we have all the condition media, cytokines, growth factors, and all of that. And on top of that, we culture the Spermatogonia stem cells. So just before seeding the cells inside the culture, we make sure that actually we're starting with the Spermatogonia stem cells. We do not have any meiotic or post-meiotic markers. So we perform immunofluorescence staining and RT-PCR to make sure that we are starting with clean Spermatogonia stem cells. I'm not going to go into details, but we success to culture the Spermatogonia stem cell and get different types of colonies inside the culture. And when we extract the cells from the agar, we did analysis to see if we can get the differentiated cells. And you can see here that using the immunofluorescence staining, we got meiotic and post-meiotic cells inside the culture. And this amazing picture, we have complete sperm inside the in vitro culture. We success to start from Spermatogonia stem cells and finish with complete sperm. This was a breakthrough in this field and a game changer, because no one did that before. And actually, we can now learn more and more about Spermatogonia stem cell. We can understand how to find a solution for men with non-obstructive azoospermia in vitro, and also understand the growth factors behind this. Actually, we published a few articles about this. We did it also with a colleague from Germany. They also did the same thing in Germany, and also they got a sperm inside the in vitro culture. Unfortunately, no one believed us, because there was a real breakthrough. It's impossible, you cannot do that. It's a very complicated process. But later on, many other success to do that in vivo and in vitro and even in human. Everything is still under clinical, is still under research. So there is a little bit of side effect that everyone afraid from it. It's to take these cells out, to culture it, and they may become cancer cells, because you do not control everything inside the in vitro. So everything is still under research, but these models help us to understand more about Spermatogonia stem cells. So as I said, we developed this model to understand the proliferation of the Spermatogonia stem cells and the differentiation. And I used to test a lot of cytokines and growth factors inside these colonies. Actually, we found that Leydig cells can release also cytokines. And cytokines actually belong to the immune system. Why Leydig cells producing cytokines? So we found that they have role actually in the immune system to protect the testes from any infections, to help the macrophages. And we found actually that cytokines, they can push or inhibit the spermatogenesis process. For example, I1 alpha, we found that I1 alpha can improve and increase the spermatogonia stem cell proliferation, while I1 beta can increase the sperm production. So it's really complicated process, but the main interesting finding that we found that testosterone block and we can say kill the spermatogonia stem cell. When we have these colonies inside the culture, if we're adding testosterone, they go through apoptosis. They're gone. So we did not understand this, why this is happening, so we keep it aside. We did not touch that. We didn't understand what to do. But later on, I will show you that actually we have proved that what we found was completely true. I will share two studies, very interesting. Study that showed that they can extract spermatogonia stem cell from non-obstructive azoospermia, and they can culture it, and these cells can proliferate and differentiate, and even they have markers for acrosome in vitro. This means that the spermatogonia stem cells inside azoospermia patient, they are good. They have the ability to proliferate, but something wrong in the testes that block these cells. We don't know why. We believe that the environment is the reason. This is why if we take these cells out, they are good, they proliferate. If you put it back, they are not proliferate. So the main thing in this study and the other one, that spermatogonia stem cells have the ability to divide again outside the testis. And the other thing, in this study, they found that all men with non-obstructive azoospermia, even the Sertoli cell only, they do have spermatogonia stem cells inside the testis. It's very difficult to find them. They are very rare, depend on each patient. But today, we have more advanced technologies to find and separate these spermatogonia stem cells. One of them is called the immunomagnetic separation. We label the spermatogonia stem cell with a specific marker on the membrane, like CK2G4 alpha. Then we label it with metal beads and put it on a magnet. And then you will have a pure spermatogonia stem cell that you can use it for study and culturing. One of the leading companies is called Military Biotech in Germany. They specialized in the immunomagnetic separation. So from these studies and other, they suggest that spermatogonia stem cells actually from men with azoospermia are healthy and can divide outside the testis. So this is the main goal from this study, and this finding will help us in the future on the next slide to develop a product. So this finding and our improved understanding of spermatogonia stem cells and have us more smarter, we know more, but there is a few pieces still missing. Now let's talk about testosterone and the role of testosterone in the spermatogenesis process. Many studies already confirmed that testosterone is very important for the late stages of spermatogenesis. This means you need it only for the sperm formation. You do not need it for in the beginning. Spermatogonia stem cells do not need testosterone. We need it only for the late stages. This has been confirmed by many research, so all of us agree about that, and I will show you also in our research how we confirm this finding. In MD Anderson lab, they also showed a new role of testosterone in azoospermic cases. Research has also found that testosterone involved in the blockage of spermatogonia stem cells in abnormal conditions like azoospermia. And the suppression of the testosterone will allow us to push the spermatogonia stem cell to divide again. Again, in 2004, we confirmed this with a different study. And in 2010, again, we confirmed that testosterone actually is the main thing that blocking the spermatogonia stem cells from dividing. I will show you here one of the studies that we did include the suppression of hormones. Actually, the main goal that we did in MD Anderson's Cancer Center, we want to understand what genes involved in the restoration of spermatogenesis. Patients that have cancer, they go through chemotherapy and radiation. Some of them, they become azoospermia for many years, and some of them recover after a few months or 1-2 years. So must be some genes that controlling this process. We want to understand and find these genes. Once we know it, maybe we can find a solution to help other men with non-obstructive azoospermia. So to do that, we start with the rat model. We call it recombinant inbred rats. You take a very sensitive rat. You can see in the left side, this rat, if you give it 5 gray radiation, it becomes azoospermic for it will not recover. And on the right hand, you see that SHR rats, you give it 5 gray, after 10 weeks it starts recovering. So we have sensitive and we have resistant rats. We do inbred between those, and we keep inbred the generation till we have 20-25 generations. What we did actually, we mixed the genes, the DNA from both rats, and then we do experiment using all the generation, collect the data using very smart software, and trying to find out the genes that are responsible for the recovery in azoospermia. Unfortunately, it became very, very complicated. We did not find that 1 or 2 genes. We found that about 20, even more genes involved in the recovery of spermatogenesis. So we did not complete the study. But what we did actually, we learned other things doing that. And one of the very important things to show you here that FSH is not the reason for azoospermia, in primary testicular failure. FSH is the results from azoospermia. Okay. This is why if we try to reduce the FSH, it will not be effective, because we're trying to play with the hormone balance, and this will not do anything. We tried on animal models, rat model, monkey model, it's not useful. The body increased the FSH because the body is feeling that something wrong and trying to fix the problem. So reducing the FSH will not solve the problem. We found actually reducing the testosterone will push the spermatogonia stem cell to divide again. On the left side, you see a section from Sertoli cell-only rats. Very nice spermatogonia stem cell. And after 10 weeks of testosterone reduction, we have 100% recovery in all tubes. As you can see here, we have only primary and secondary spermatocyte. We do not have sperm. Why we do not have sperm? Because we do not have testosterone. Once we remove this reduction, the cells can complete the cycle and produce sperm. Again, we also showed here that testosterone is very important only for the late stages. Another interesting finding that the number of the spermatogonia stem cell control the recovery process. The more spermatogonia stem cell we have, the recovery will be much faster. So the lower spermatogonia stem cell we have, the recovery will be very long. This can be related also to FSH level. And different study they showed when you have very high level of FSH, this means you have a low number of spermatogonia stem cells. When you have 30, 40 FSH, this means the recovery will be much slower and can take a year, maybe more than a year. So we can summarize the studies that testosterone blocks spermatogonia stem cell in abnormal conditions, and the suppression of testosterone will push the spermatogonia stem cells to divide again. These findings improved actually our understanding about testosterone and the relation to the spermatogonia stem cells. We now know more and more about spermatogenesis process. And this study has been done in the Marvin lab in the Anderson Cancer Center with Professor Guna. They spent about 34 years investigating, at least Marvin, investigating this. But unfortunately, nothing it's all basic science, nothing come to clinical. So one day I asked Marvin why we don't go to clinical study, why we don't try it on human. So he said there's no enough data about that, and we try on radiation therapy patient and it was not successful because maybe the radiation killed the spermatogonia stem cell. So I decided to take a step and open a startup company called Midhaija to do research and develop treatment and that based on a lot of knowledge that we have. We collect a lot of data, a lot of knowledge, why we don't use it. This was the main idea from Midhaija. I get support from family and friends, and we start looking for fast solution. So to do fast solution, we try to look for ingredients and that are from natural source. We do not do any genetic modification, and looking for these ingredients that can actually reduce the testosterone level, and there is no side effect. And the outcome was Prolistem, and Prolistem founded in 2012. Based on the research, we got patent on that in 2017. It's based on 100% natural ingredients, active ingredients. We did not do any changes to these molecules. And it's the main thing, it's a new treatment strategy. No one used it before. And this was the main goal from creating Prolistem, based on a lot of this outcome research. So it's designed to support non-obstructive azoospermia, and I must say for primary testicular failure. It's not for secondary testicular failure, it's only for primary testicular failure. And it's called in a unique mechanism that called spermatogenesis restarting process. What I mean by that, if you have a problem with your smartphone or your computer, you do restart for the device, and in 80% of the cases it will solve the problem. Okay? So this is the idea. We do restart the process of spermatogenesis. When you reduce the testosterone, we do not know exactly what is the mechanism. We believed when you reduce the testosterone, actually you change the function of the Sertoli cell only, and changing this function for a period of time, it will give the spermatogonia stem cell the opportunity to start dividing again. And because we do it in natural way, so we reduce it in 10-20%. And it must be for a long time, like 3 months, because spermatogonia stem cells division is very, very slow. It takes about 24 days for the spermatogonia stem cell to divide. So the Prolistem 6 months treatment, the first 3 months, we call it phase 1. We do testosterone reduction, okay? And hopefully the spermatogonia stem cell, they divide in the first 3 months. We move to phase 2, testosterone increase, to try and push the cells to complete the cycle and produce complete sperm. Again, stage 1 and stage 2 from Prolistem block testosterone production inside the testes, and I will show you how we do it. I cannot go into details about the ingredients, but I have been asked the main ingredients. It's from licorice, and it's called glyceric acid, and it's known that it's black enzyme in the pathway of testosterone production. It's the 17 beta dihydrosteroid dehydrogenase, and when you block this enzyme, actually you do a reduction for the testosterone level. Again, we cannot do 100% reduction, it's about 10-20%, but it's good enough to push the spermatogonia stems again. And we have more ingredients that showed in our rat models that also reduce the testosterone level. A combination of these ingredients give us the effect that we want. I want to show this slide because Muhammad asked me about this acid. This acid is very safe. It's approved by the FDA. Of course, it's like other ingredients. If you take it in high dose, it can have side effect on people with high blood pressure. Exactly like all the vitamins that we know, A, D, E, carnitine, everything. If you take it in high dose for a very long time, it will have a side effect. So it's very safe, it's FDA approved, it's in the list, and the doses that we are used are controlled by the FDA. So after the development of Prolistem, we tested on animal model first to see if we can get really the effect that we want, and after 2 months we showed that spermatogonia stem cells start to divide again. Of course, we cannot get the same effect that we got in the rat model that we showed before, because in that model we reduced the testosterone to 0. Here we cannot do that using the natural product, but we success to push the spermatogonia stem cells to recover again, and we found complete sperm inside the testes after 1 and 2 months. Then we did a small clinical study to understand if we can achieve also this effect in a human. You know, a human is different from rats. In rats we got 100% success. In a human we cannot get that. There is a lot of difference between people, genetic reasons, and all of that. So we did a study in 89 patients. We exclude the genetic issues. Everyone with genetic or family history, they did not perform the study. And here I'm showing a few examples that testosterone before and after stage 1 and 2, you can see that there is a reduction of 10-20% in testosterone. After the 6 months treatment, we found sperm in 43 patients. 21 patients we found sperm in the semen. It was between like 10 sperm to few millions. And in 22 we found sperm in biopsy. We did not success to find any sperm in 46 patients. All of them they did a microtesting, but there is no sperm found. Some of them was sertolizing, only maturation arrest or mixed. Unfortunately, many of them we don't know the histology, because many doctors actually don't send to histology, just say there is no sperm and that's it. We continue doing clinical study because, as I said, the more study we do, the more clear picture we will see and we will have. This is another study that have been done in hospital in the third party. All the 54 patients, the non-obstructive, they all negative in the biopsy. All of them did biopsy before. 28 patients did not get Prolistem, and we found 21% success by microtesting. All of them they went through microtesting. 26 from the patient they took Prolistem for 6 months, and the success rate increased from 20 to 50%. This is confirmed also our previous one, because in the previous one we showed that 23% found sperm in semen and 25% find sperm in in biopsy. Just to align everyone about the difference between supplement and drug in the United States and the FDA. Drug is a chemical medicine. Supplement is taken from natural ingredient, and you cannot do any changes. Once you do change, it becomes a drug. And the regulation is different between drugs and supplement, because drug must be tested in clinical study. And why they test it? Because the more important thing is to see the side effect of these drugs. The success rate of drugs can be 10, 20, 30%, but the more important thing is the side effect. It must be safe. You cannot give a drug to patient and cause a different damage. So this is why it's important to go through phase 1, 2, and 3. It can cost about $100 million, maybe more. Very complicated. This is why also we choose to go in the natural way. And as I said, both of them regulated by the FDA, but supplement does not need to do pre-market approval. You must manufacture in GMP facility and FDA approved facility. Drugs, they must be pre-approved before enter the market. A few things about azoospermia management. This is from the guidance from the Canadian and the United States. The best solution for men with primary testicular failure is to do the microtesting. Okay? And they said there is no data support the hormone treatment for men with primary testicular failure. Actually, they have a lot of hormones. I think they do not need to have more hormones. They have a high level of FSH or normal level of FSH. They have normal hormones, levels of testosterone and LH. So hormone treatment actually is like putting water on the flowers in raining day. So you just increasing the hormones. And very interesting finding that 9% of the men, they may have sperm inside the testes or in the semen, but we miss to find them. Why? Because if you have thousands of tubules, they are not good, but you have few of them, 10% or 5%, they have the ability to produce sperm. And if the man go and did semen analysis today, they may not find anything, but if he did after a few weeks, he may found sperm. And this a lot of cases happening with me and with Muhammad. After the treatment, he went and gave a semen analysis. They found a few sperm. They did not freeze it. He came back after 1 week, no sperm. After 1 month, no sperm. So it was very difficult to find the point when we can get the sperm. So we are developing we're developing a solution to try and solve this problem and save, just to find the point where we can get sperm. It's called a sperm sure. It's a collection kit that the patient will do a semen collection every week for 3 months. It's a long procedure, but it can save him a lot. And after the collection, with a special kit that can keep the sperm inside, there is a special membrane reagent that we developed. He will send it to us in Houston labs, and then we will do the analysis and send him a report telling him what is the best time to do the semen analysis or to do the microtest to increase the chances to find. It will look like this. We fix the sperm on the filters. This is an example how it's look like. And a few things about and recommendation, of course. Repeating the microtesting will reduce the chances to find sperm the next time. This is why we highly recommend to increase the chances of the patient before going to the microtesting or testing, to give the treatment 6 months before and then do the microtesting. We want to increase his chances as much as we can. And if we did a microtesy and we want to repeat it, it's recommended by the guidance to wait 6 months before repeat the microtesy for full recovery. Again, I talked about the level of FSH. There is a lot of studies and it's very difficult really to understand what is the best level of FSH to do the microtesy. But let's say high levels of FSH like 30 and 40 indicating severe problem, and the recovery will be very long. And recently there is an article published about the equipment that we use during the microtesy. There is the optical and the microscope will highly increase the success chance to find sperm during the microtesy. Okay, genetic is a big problem. More and more genes discovered that responsible for azoospermia. As I said, in our research in MD Anderson, we found almost all the chromosomes involved in the spermatogenesis process. But the main test that we can do today is the AZF factors. And you can see here that if there is a deletion in A, the chances will be 0. If there is a deletion in B, the chances also will maybe 0, depending on the deletion in what deletion. And of course, if it's C, there is a big chance to find sperm inside the testes. So genetic tests are really recommended to do it, the karyotype and the AZF factor. And we all the time telling the patient, if there is a genetic problem, the success chances will be really low. Okay, we are very clear about that. And even if he did the AZF factor and the karyotype and everything is okay, we always asking about family history. If you have family history, brother, uncle, it will indicate genetic issue and will reduce the success chances. So, our recommendation for taking Prolistem, to start with, of course, we know all of them azoospermia, they don't have any sperm in the semen, so we start with the hormone levels. 99%, if they have high level of FSH, they have non-obstructive azoospermia, primary testicular failure. In this case, before going and opening the testes by any surgery, it's highly recommended to increase the chances to the patient before doing that, because as I said, reopening all the time will reduce the chances. So we don't have a problem with high FSH. High FSH means primary testicular failure. When we have normal testosterone, LH and FSH, now it can be obstructive or non-obstructive. Even the ultrasound may not tell us if it's obstructive. If any small biopsy is needed to confirm if it's obstructive or not. If it's non-obstructive azoospermia, then it's highly recommended to take the treatment 6 months before the microtesy. We hope, of course, when we do the biopsy to find few sperm, and then we save the patient from doing the microtesy. If there is as I said, if it's secondary testicular failure, there are problem with the hormones, FSH, LH, Prolistem cannot help in these cases. Okay, and if we have a problem with the testosterone, with the Leydig cell failure, also Prolistem cannot help because it's not a hormone treatment. So our vision to become the gold standard of azoospermia treatment worldwide, and to be recommended to be taken before doing the microtesy. It's a new strategy treatment. It's different from the traditional thing, and based on all the data and the knowledge that I showed to you and all the clinical study, we believe it's the best solution that will give the patient a high success rate before doing the microtesy. So our mission is to deliver happiness to families. When we receive these messages all the time, it will make me and really happy. This is the important thing. I believe it's a fingerprint that we can live in this world. And I hope, as I said, to become and really help a lot of families using this treatment.